Good day to everyone. I'm Dr. Li Zixue, lecturer of University Technology Malaysia (UTM), the moderator of the forum Good Night. So first of all, I would like to introduce our panelists in our forum. So our first panel is Dr. No Ishua Binti Wahidi, the lecturer of International Medical University (IMU). The second panel is Dr. Su Sing Gee, lecturer of University Kebangsaan Malaysia (UKM). And our final lecturer, Dr. Kuganeswaran, lecturer of University Putra Malaysia UPM. So, for today, our forum will discuss about the application of genetic engineering in pharmaceutical field. So, first of all, what is genetic engineering? So, genetic engineering was discovered by American biochemist Dr. Stanley Cohen and also Dr. Herbert Boyer in 1973, where they try to cut the DNA into the fragment and then rejoin this fragment into the different fragment and then transform this fragment into the bacterial cells to reproduce in large amount. So this is the first technique that used in genetic engineering by these two scientists. So in pharmaceutical field, the first drug product produced was insulin in 1978 by David Goddard. So this drug insulin already gives many beneficial, especially to the diabetes mellitus patients. So from the first drug produced insulin, so nowadays many scientists also use this technology, genetic engineering, to apply and produce drug product that can contribute in pharmaceutical industry. So without wasting our time, I would like to invite our first panel, Dr. No Ishua Binti Wahidi, the lecturer of IMU, to give a speech in introduction of genetic engineering in pharmaceutical fields. So the stage is yours. Dr. Ishua, the stage is yours. Thank you.
Thank you, Mr. Lee Jizwan, our moderator of the day. Genetic engineering has provided a way to create new pharmaceutical products called recombinant DNA pharmaceutical. Such products include antibiotic, drugs, vaccine, and hormone used to treat various diseases. Genetic transformation studies have shown the potential of producing recombinant proteins, including pharmaceuticals and industrial proteins and other secondary metabolites in plants. Several substances have already been produced in transgenic plants or in different stages of clinical trial, but none of them were approved as pharmaceutical for humans until 2012. Recently, an enzyme for treating the rare hereditary Gaucher diseases generated in carrot tissue became the first plant-made pharmaceutical for human used to gain regulatory approval by the US FDA. Genetic engineering is also used to manufacture subunit vaccine, which are safer than the other vaccine because they contain only a single antigenic molecule and lack any part of the genome of the pathogen. For example, a vaccine for hepatitis B is created by inserting a gene encoding a hepatitis B surface protein into a yeast. This yeast then produces this protein, which the human immune system recognizes as an antigen. The hepatitis B antigen is purified from yeast cultures and administered to patient as a vaccine. Even though the vaccine does not contain the hepatitis B virus, the presence of the antigenic protein stimulates the immune system to produce antibiotics that will protect the patient against the virus in the event of exposure. Genetic engineering has also been important in the production of the therapeutic proteins such as insulin, interferons, and human growth hormone to treat a variety of human medical conditions. For example, at one time, it was possible to treat diabetes only by giving patients pig insulin, which caused allergic reaction due to small differences between the proteins expressed in human and pig insulin. However, since 1978, the recombinant DNA technology has been used to produce large-scale quantities of human insulin using E. coli in a relatively inexpensive process that yields a consistently effective pharmaceutical product. So I hope this short and brief introduction can give a picture to all about what does this in genetic engineering contribute and the importance of genetic engineering in pharmaceutical field. Thank you.
Thanks, Dr. Inshira, for your introduction about the genetic engineering in pharmaceutical space. So now, what I think everyone will something know about the history and the tips of genetic engineering in pharmaceutical field. So now, it will be climate in our forward, which we will share about the current application used in genetic engineering in pharmaceutical field. So without wasting our time, I would like to invite Dr. Su Singyi, lecturer of UKM, to give her speech about the production of interferon from genetic engineering. So Dr. Su, it's your turn now.
Thank you, our moderator. Hi, my name is Singhi. I'm a lecturer from UKM. Now I will talk about the application of genetic engineering in pharmaceutical sector. Genetic engineering has a great contribution in pharmaceutical use. It helps in the production of drugs and hormones. The earliest users of genetic engineering in pharmaceutical were gene splicing to manufacture large amount of insulin, which was made by using cells of E. coli bacteria. Insulin is a protein hormone which produced in the pancreas and is important in the blood sugar level regulation. The function of insulin is to facilitate the transport of glucose into the cell. Insulin deficiency can cause diabetes mellitus or sugar diabetes, in which the blood sugar level will rise, resulting in harmful consequences. Research shows that at least 3% of the world population is suffering from diabetes mellitus, and the sufferer require insulin injection to manage the disease. Before genetic engineering, the pancreas of slaughtered pig and cattle were the main source to produce insulin, which is used in treatment. However, this source of insulin has minor difference in the amino acid composition compared to the insulin produced in humans. Moreover, insulin which is obtained from animals also contain trace impurities. As a result, some patients were allergic to insulin sourced from animals and had damaging side effects after getting the injection. This problem was able to solve by inserting a gene code for the insulin in yeast or bacterial cell. Next, interference, which is also a product of genetic engineering, is used to eliminate certain viruses and kill cancer cells. Interferon are proteins of human natural defenses. Commercially available interferons are human interferons manufactured by using recombinant DNA technology. The mechanism of this action of interferon is quite complex. Interferon help in modulating the responses of immune system to viruses, cancer, and other foreign substances that invade our body. They do not directly kill the virus cells, but boost the immune system to respond and reduce the growth of cancer cells. This can be done by regulating the action of several genes that control the secretion of numerous cellular protein that affect the growth of cancer cells. Genetically engineered type of interferon is used to treat a rare form of adult leukemia. The recombinant DNA technology takes genes that contain the codes for interferon and combine them with harmless bacteria. The bacteria then become a host cell, which is capable of manufacturing large amounts of interferon identical to that produced by the human body. So with that, that's all from me. And now I'll pass it to the moderator. Thank you.
Thank you, Dr. Su, for a nice sharing to us about the interferon production using genetic engineering. It is interesting to me, and I also first time heard about it that the interferon can be produced by using genetic engineering. So last but not least, the last panel of our forum today, Dr. Kuladeswaran, lecturer of UPM, will give a talk about the human growth hormone production by using genetic engineering technique nowadays. So, Dr. Kugan, you can share your opinion now. So, your stage is yours.
Thank you to our moderator Lizzie Swan. My name is Kuhaneswaran son of Suresh. Now, let us have a look on how genetic engineering have contributed to the production of human growth hormone, which is also known as HGH. In ancient times, animal tissues have been used as a medicine. Even in the 20th century, the human growth hormone, which is extracted and purified from our pituitary gland, is still used as a medicine to treat growth deficiency. Since the genetic engineering of host cell become possible, the whole new medicine using recombinant DNA technology has emerged. This medicine from genetic engineering is called biopharmaceuticals. One of the prominent example of biopharmaceutical is human growth hormone or HGH. Naturally, human growth hormones is produced by our pituitary gland, which is a pea-sized organ in our brain. It helps with variety of functions. This human growth hormone has two main functions. The first one is stimulating growth during our childhood, and the second one is impacting our metabolism. But sometimes there will be a shortage of this naturally producing human growth hormone inside our bodies, which resulted in stunted growth in children. Thus, to overcome this, the US Food and Drug Administration has approved the production of synthetic human growth hormone. So when we talk about synthetic human growth hormone, this is where genetic engineering technique comes in. This is because we use genetic engineering technique to mass produce synthetic human growth hormone, which is also known as recombinant human growth hormone. As an overview, by using genetic engineering technique, we genetically modify an organism, usually prokaryote like E. coli, with, by inserting a gene encoding for human growth hormone gene. And this genetic modification will turn the E. coli cell into a little factory that can mass produce our desired human growth hormone. This will lead to limitless source of human growth hormone with less risk of contamination. Now, what are the uses of this genetically modified human growth hormone? First of all, it can be used as a treatment for people suffering from growth, for children suffering from growth deficiency. Secondly, it can be used to treat people who are suffering from Turner syndrome, chronic renal insufficiency, and also renal carcinoma. And thirdly, it can use to increase the milk production in lactating cows and also to increase the muscle mass of cattle. That's all for me. Thank you.
Wow, it's fantastic and interesting knowledge to us. So thanks again for Dr. Kugat. So that's all, and we near the end in for our forum. So on behalf of UTM, the lecturer of UTM in Department of Bioscience, and also the behalf of university, I would like to give the highly appreciation and thank you to the panelists that can attend today. So thanks you guys can spend your extra time to attend the forum today and then share the interesting knowledge to us and our students about the genetic engineering in pharmaceutical field. So that's all for our today. So thank you very much to all panelists and also students that can attend our forum today, that you spend our time to listen the panelists talks and what the panelists will do. So hopefully the knowledge, the talks that the panelists give can beneficial to use in your future and give the extra knowledge to you. So that's all for our for our forum. Bye, and we see you again. Bye.